Welcome to today's deep dive. I want you to imagine, just for a second, that you are crawling exactly 145 meters deep into the Earth.
Oh, wow. Yeah, that is not for the claustrophobic.
No, definitely not. You are navigating these treacherous, completely pitch-black passages of the Rising Star Cave System in South Africa. The air is thick, the spaces are agonizingly tight-like, some squeezes are barely wider than a ruler.
Just thinking about that makes my chest tight.
Right. But, okay, at the end of this grueling journey, you discover something breathtaking, a massive trove of fossils from a mysterious human relative known as Homo naledi. And these date back roughly 300,000 years.
Which is just a massive, unbelievable find.
Exactly. Now, naturally, you'd assume what, any archaeologist would assume upon finding a huge group of bones. You'd think, okay, this is a standard mixed demographic of individuals who just somehow met their end together in this dark chamber.
Right, like a random cross-section of a population.
Yeah. But what if a brand new molecular test just proved all those basic assumptions entirely wrong?
I mean, it completely flips the script on what we thought we knew about this site.
It really does. So our mission today is to unpack this groundbreaking paper from July 2026, published in the journal Cell. We are going to explore every single detail of how scientists used cutting-edge protein analysis to uncover a logic-defying demographic secret about 20 of these extinct individuals.
It's honestly one of the most fascinating pieces of paleontology work I've seen in years.
Before we get down to the molecular level, though, let's set the stage for you.
Oh.
Who were these Homo naledi individuals in the first place?
Well, Homo naledi is this incredibly fascinating Middle Pleistocene hominin. They're the textbook definition of a mosaic species.
A mosaic species,
Yeah.
like a mashup of different traits.
Yeah, exactly like a mashup. If you looked at their brains, their shoulders, and their trunks, you would see features that are very small and, well, ape-like.
Right.
They look a lot like much earlier hominins, like Australopithecus, swinging through the trees. But then you look at their faces, their hands, their feet, their lower limbs.
And they look completely different.
Yeah, they look remarkably Homo-like, much closer to us, like they were built for walking upright and manipulating objects.
So they really challenged that old visual we all grew up with, right? That straight line march of human evolution from a hunched ape directly to a modern human.
Oh, completely. Evolution is much more of a tangled bush than a straight line.
But one of the most famous things about the Homo naledi fossils found in the Rising Star Caves is how physically similar they all look to one another. Like, they are incredibly homogeneous in size and shape.
Yeah, the morphological variation is extraordinarily low. And for a long time, researchers have been debating why that is.
Like, is it just a trait of the species as a whole? Did all Homo naledi just look identical?
Right. Or does it hint at something deeper? Maybe something genetic or even cultural about the specific individuals gathered in this one particular cave. And that is exactly the mystery this new chemical analysis was designed to solve.
Okay, so here's my first big question. If you want to know who a 300,000-year-old individual was, why can't we just, swab the bone, extract some ancient DNA, and run a sequence?
Ah, the CSI approach.
Yeah, we see it on TV all the time. They put a bone fragment in a machine, and 10 seconds later, they have a full genetic profile. Why not just do that?
Well, it works beautifully on TV. And to be fair, it occasionally works in real life if a specimen has been frozen solid in permafrost for millennia.
Like a woolly mammoth or something.
Exactly. But DNA is a highly fragile molecule. It degrades over deep time through a process called diagenesis.
Diagenesis. Okay, what exactly is happening there?
Well, in a warm, humid climate like South Africa, the ambient heat and the environmental moisture literally break down the phosphodiester bonds.
The bonds holding the DNA together.
Right, the bonds that hold the DNA backbone together. So over hundreds of thousands of years, those long strands of genetic code, they just completely fall apart. The slate is wiped clean.
Wow. So the heat just melts the code away. But if the DNA is entirely gone, what's left for scientists to actually test?
Proteins. And this is where we enter the really cool realm of paleoproteomics via mass spectrometry.
Paleoproteomics, so studying ancient proteins.
You got it. Proteins are essentially chains of amino acids, and structurally speaking, they are much hardier than DNA.
Hardier how? Do they just take longer to break down?
Yeah, especially when they are locked inside dental enamel. I mean, enamel is the hardest, most mineralized tissue in the human body.
Okay, so the teeth are acting like a vault.
Exactly like a vault. It's a closed, heavily armored time capsule. It traps these ancient proteins safely inside its crystalline structure and protects them from the outside environment.
Okay, but that brings up another problem. How do you get the proteins out of a priceless, one-of-a-kind fossil tooth without just destroying the tooth. I mean, in the past, scientists literally had to take a tiny saw and cut a chunk of the tooth off.
Yeah, they did. It permanently altered a piece of human history, which is heartbreaking for a museum curator.
Oh, totally. But for these fossils, they used a technique called acid etching. And I want to give you an analogy to help picture this.
Oh, I love a good analogy. Go for it.
Imagine taking a delicate frosted cookie and gently dipping it into a glass of milk for just a few seconds. The milk dissolves a tiny microscopic layer of the sugar on the surface, but the cookie itself stays completely intact.
That is exactly what they did, just with slightly more dangerous chemicals than milk.
Right, a bit more intense.
They took the Homo naledi teeth and dipped them in a 10% solution of trifluoroacetic acid.
TFA.
Yes, TFA. And they only left it in for about 10 to 15 minutes. It microdestructively solubilizes the peptides, which are the short chains of amino acids, right at the very surface of the enamel.
Just skimming the frosting off the cookie.
Precisely. And the truly astonishing part of this study is that this gentle microdestructive etching actually worked better. It yielded far more data than taking a saw and cutting the tooth.
Wait, really? A quick acid dip gave them more biological data than physically grinding up a chunk of the tooth? How does less destruction give you more data?
It comes down to chemical exposure time. In their initial tests, the traditional destructive cutting yielded about 12,243 peptide spectrum matches.
PSMs.
Right, PSMs. That's essentially the number of protein fragments the mass spectrometer confidently identified. But the acid etching, that yielded 18,709 PSMs.
That's a huge jump. Why the difference?
When you demineralize a cut chunk of enamel, the ancient proteins have to sit in a really harsh acid bath for about 48 hours to break down the chunk.
2 whole days in acid.
Yeah, and that prolonged exposure causes terminal hydrolysis. The acid actually starts chewing up and destroying the very peptides you're trying to study.
Oh, so it's destroying the evidence while trying to extract it.
Exactly. But with the etching technique, the peptides are in the acid for less than an hour. So they emerge longer, more intact, and just way more informative to the mass spectrometer.
Okay, I get that. But, and here comes my skepticism, if you're just washing the outside surface of a tooth, how do we know these aren't just modern proteins?
What do you mean?
Like, what if an archaeologist sneezed on the tooth back when it was excavated in 2013, and the machine is just reading their modern lunch?
It's a great question, and honestly, authenticity is the biggest hurdle in paleoproteomics. But the mass spectrometer doesn't just read the sequence of amino acids. It is sensitive enough to detect their chemical age.
Their chemical age. So there's like a biochemical fingerprint of time.
Exactly. Over deep time, amino acids undergo spontaneous chemical modifications that simply cannot be faked or produced by a modern sneeze.
Okay, tell us exactly what changes occur to prove these proteins are actually 300,000 years old.
So there are 3 major chemical markers. First, there's deamidation. Over millennia, the amino acid glutamine spontaneously drops an opide group and turns into glutamic acid.
Okay, that's one.
Second is the conversion of arginine into ornithine. And the third, and perhaps most elegant, is racemization.
Racemization. Let me try to break that down. Amino acids come in 2 structural shapes, right? L and D, which are basically mirror images of each other.
Yes, exactly like your left and right hands.
And in living things, almost all amino acids are held in the L shape. But after death, they slowly undergo this chiral switch and literally flip to the D shape.
You've got it.
So a good way to remember it. L for living, D for dead. That is such an elegant way to track how long something's been gone.
It really is, and the Homo naledi samples showed advanced levels of all these deep time modifications, heavy deamidation, high levels of ornithine, and significant D-amino acid conversion.
So the chemical proof is undeniable.
Completely. These proteins are genuinely ancient. They originated from the hominins themselves, not a contaminated sneeze.
Okay, so let's do a quick recap of bullet point number 1 before we move on. First, DNA fails over time due to heat. Second, proteins survive much longer inside the enamel vault. And third, a 10-minute acid etching saves the fossil while yielding better data, authenticated by molecular aging.
Perfect summary.
So I want to ask you, the listener, if you had a chemical swab that could extract the entire biological history of any fragile ancient artifact in the world without breaking it, what would you test first? I mean, every museum collection on Earth is now a potential gold mine of new data.
It opens up a whole new frontier. But the primary goal for this specific team wasn't just to prove their cool new extraction method worked.
Right, they had a bigger mission.
Yeah, they wanted to use those proteins to solve the demographic mystery of the Rising Star Cave. They specifically wanted to determine the biological sex of the individuals piled up in the dark.
Okay, but how exactly does a protein sequence tell you if someone is biologically male or female?
We look at a specific family of proteins found in the enamel called amelogenins. The gene that produces this protein is located on the sex chromosomes.
Okay, so the X and Y chromosomes.
Right. We have MLXX, which is expressed from the X chromosome. Because both biological males and females have at least one X chromosome, everyone produces AMLX.
Everybody has the X, but then there's Amelie.
Exactly. Amelie is expressed exclusively from the Y chromosome. So ordinarily, Amelie is only found in biological males.
Okay, so if you find Amelie, you have a male.
Yes. Now, it's worth noting that Amelie is usually expressed at a much lower rate, roughly 10% the rate of AMLX. But if a Y chromosome is present, the Amelie protein is definitively there in the enamel.
And the mass spectrometer is basically this high-tech sorting machine, just hunting for the minute differences between those two proteins.
Basically, yeah. The sequences of human AMLX and Amelie differ by 23 diagnostic amino acid substitutions. But the absolute gold standard marker the machine is hunting for is a very specific methionine insertion at position 59 in the Amelie sequence.
Position 59.
Right. If the mass spectrometer detects that methionine in that exact spot, boom, it confirms the presence of a Y chromosome.
Okay, here's where my brain gets stuck, though. If they scan a 300,000-year-old tooth and they don't find that Y chromosome protein, couldn't that just mean the sample was too degraded?
The classic absence of evidence problem.
Yeah. Like, how do you prove a negative? How do you know the machine didn't just miss a super faint signal buried in the background noise?
And that was the central challenge for the researchers. They had to prove to a highly skeptical scientific community that an absence of a signal was a biological reality, not a mechanical failure.
How do you even prove that mathematically?
By building a rigorous statistical framework. First, they applied a base 10 logarithmic transformation to the raw, highly skewed mass spectrometry data.
Okay, wait.
This gives you a normal homoscedastic distribution.
Whoa, hold on. Homoscedastic. You're going to have to translate that for me before we move on.
Fair enough, fair enough.
You just mean smoothing out all that messy, wildly fluctuating data so it becomes a symmetrical, predictable bell curve, right? Where the variance is equal across the board.
Exactly. You turn the chaos into a clean bell curve. Once you have that normalized curve, you can apply standard, robust statistics.
Okay, I'm with you.
From there, they establish what's called the lower limit of quantitation, or LOQ.
LOQ. What is that?
It's the exact signal strength where the machine stops reliably detecting peptides. Anything below that limit is just noise. Then, they looked at known male hominin fossils to establish a baseline.
Which fossils did they use?
They tested teeth from a male Denisovan and a male Homo antecessor. By doing this, they measured exactly how strong the baseline MLX signal has to be to guarantee that the much fainter Amelie signal pushes above that lower limit of quantitation.
Let me try to visualize this. Imagine you were at a loud rock concert, and I tell you to listen for a pin dropping on the floor.
You'd never hear it.
Right, you can't hear it. But you also can't prove the pin didn't drop, because the baseline background noise is just way too loud.
Exactly.
But if we are sitting in a perfectly silent library, and I tell you to listen for a pin dropping, and you hear absolutely nothing, you can mathematically prove that no pin was dropped, because the conditions were perfect to hear it.
That is a perfect analogy. The researchers generated a z-score, calculated standard deviations, and essentially proved that the MLX signals, which is the baseline volume in your library in these Homo naledi teeth, were so incredibly strong and clear that if the Amelie pin drop existed, the machine absolutely would have detected it.
Wow.
Yeah, the statistical proof is ironclad.
Okay, so to recap our second big bullet point, we know exactly what the male protein looks like. The methionine at position 59, and rigorous math proves the machine didn't just overlook it due to degradation.
Spot on.
So think about how often in life we have to rely on the hazy absence of evidence. I mean, having statistics actually prove that an absence is a concrete, undeniable fact sets the stage for a massive revelation. So what did they actually find in the cave?
Well, out of the 20 individuals tested, which spanned 23 different tooth samples, they found 0 male markers.
0.
0. 19 of these individuals were definitively female, with over a 95% probability. And the last one was highly consistent with being female.
A completely female and juvenile sample buried deep underground. That is staggering. And it completely upends years of assumptions, specifically regarding a very famous specimen from the cave nicknamed Neo.
Oh, Neo. Yeah, that was a big shock.
Right, because for years, paleontologists looked at Neo's skull, which is relatively robust, with a noticeably larger cranial capacity, and just naturally assumed, okay, this is our big adult male of the group.
It's a classic case of morphology and our own inherent biases leading us astray.
Because we just assume bigger means male.
Exactly. We often assume larger equals male when looking at primate fossils, but the proteins don't lie. The mass spectrometer proved that Neo, the presumed robust male, is actually female.
Okay, but let's talk odds. If you have 20 individuals randomly fall into a cave over time, what are the actual odds they all just randomly turn out to be female?
If we assume a standard 1-to-1 sex ratio in the living population, the odds of randomly accumulating 20 individuals and getting all females is roughly 0.00054%.
Wait, read that again, 0.
0.0000954%. It is essentially a statistical impossibility for this to be a random accidental accumulation.
Okay, but what if it's a biological quirk? Like, is it possible that Homo naledi males simply evolved a mutation to delete the AMA gene entirely?
You mean if they didn't have the gene, they wouldn't produce the protein?
Right, and then every single male would just look female to the machine.
It's a smart thought, and we do actually see AMLI deletion in rare modern human populations.
Oh, we do?
Yeah. For example, it occurs at a rate of about 0.018% in Austria and up to 8% in certain Pakistani populations with no negative health consequences.
Okay, so it's possible.
It's possible for individuals, but a population-wide deletion across an entire species, given how closely their other proteins mirror ours, it's incredibly unlikely. In science, we rely on parsimony.
The simplest explanation is usually the right one.
Exactly. The simplest and most logical explanation is that the Y chromosome markers are missing because males are missing. They're actually all females and juveniles.
So, time for recap number 3. We have a 0% male finding, the dethroning of the male king Neo. So I want to ask you, the listener, what makes more sense to you? A bizarre genetic anomaly where all males lost a Y chromosome protein, or a hominin species intentionally carrying only their mothers and children into the dark?
That really is the million-dollar question, isn't it?
It really is, because we have a cave entirely filled with women and children. Why? Is it purely logistics? Like, the cave passages are so tight, maybe only the smaller bodies of females and kids could physically squeeze in there to explore or hide from predators.
That's the logistical theory. But experimental archaeology shows that the passages, while incredibly tight for a modern human, wouldn't have been an absolute barrier for an adult Homo naledi male.
Because they weren't that much larger than the females to begin with.
Exactly. Which leads us to the more controversial and frankly breathtaking hypothesis: deliberate, sex-segregated cultural mortuary practices.
You mean they were intentionally carrying only their mothers, daughters, and young children deep into this terrifyingly dark cave to intentionally bury them?
That is what the data seems to suggest.
But that implies a level of socio-cultural complexity, grief, and ritual that is shocking for a species with a brain a third the size of ours.
It forces us to completely rethink what small-brained hominins were capable of. But, and here's where it gets even crazier, the proteomics had one more massive shock in store for us that adds another whole layer of tragedy to this group.
Wait, there's more.
Oh, yeah. It wasn't just their biological sex that was identical.
What else was identical?
Across all 20 individuals, there was 0 variation in the recovered protein sequences. None.
Wait, 0 variation?
None. They were essentially molecular clones across the 509 amino acid positions the researchers could confidently map.
Is that normal? I mean, if you grab 20 modern humans off the street today, would their proteins be identical?
Not a chance. The researchers actually ran computer simulations to test this exact scenario using the 1000 Genomes Project.
Okay, what did the simulations show?
They simulated grabbing 20 random people from modern populations 1,000 different times. In diverse modern African populations, the probability of finding 0 variation in 20 people is exactly 0. It never happened.
Never.
Never. And even in highly isolated modern populations, the chance of finding 0 variation was only between 1 and 12%.
Wow. So if they are this genetically identical, it means they weren't part of a vast, thriving, diverse population.
Right.
This implies that this specific group of Homo naledi suffered from prolonged isolation and intense inbreeding. Like they were a deeply closed-off family unit.
Exactly. But hidden within that identical code were two specific evolutionary breadcrumbs.
Oh, I love a good breadcrumb.
These are single amino acid polymorphisms, or SAPs.
SAPs.
Right. They are tiny single-letter changes in the protein code that help us map exactly where Homo naledi sits on our family tree.
Okay, let's follow the breadcrumbs. What do these mutations tell us?
So the first one is in the AMALX protein at position 141. Humans, Neanderthals, and Denisovans all have the amino acid tyrosine at this spot. But Homo naledi has phenylalanine, represented by the letter F.
Okay, and F. Where else do we see that?
This F trait is uniquely shared with ancient primates, specifically lemurs and certain monkeys.
So they have an F at position 141, just like lemurs.
Yep.
And what's the second breadcrumb?
The second is in a collagen protein called COL17A1 at position 635. Modern humans have alanine, but Homo naledi has proline, represented by a P.
A P. And who shares that?
This P is an ancestral trait shared with Paranthropus robustus, which is a robust ape-like hominin.
Okay, so let's make a mnemonic out of this. F for furry lemurs, P for Paranthropus.
That works perfectly.
It paints such a clear picture of this mosaic hominin at a molecular level. I mean, they have the F and the P tying them directly to ancient apes while physically having hands and feet that look just like ours.
They are a literal bridge between the ancient and the modern, trapped in their identical genetic code.
So, recapping this final section, extreme inbreeding, zero genetic variation, and a mosaic code bridging ancient apes and humans. And I want to ask you, listening to this, does knowing how isolated, severely inbred, and genetically identical this group was change how you view their mysterious gathering in that cave?
It definitely changes it for me. It paints a deeply poignant picture of a struggling population, perhaps the tragic dead end of a family line, just retreating deep into the earth.
We started this journey with a tiny drop of acid, gently dissolving the invisible surface of a 300,000-year-old tooth. And from that single drop, algorithms and mass spectrometry completely bypassed the limits of degraded DNA.
It's amazing what technology can do now.
Right. They uncovered a highly inbred, completely female and juvenile population of ancient human relatives harboring ancient ape genetics, hidden 145 meters underground.
It really proves we can extract vast amounts of data without destroying the fossils we are entrusted to protect. And more importantly, it forces a complete paradigm shift regarding the social dynamics and the intelligence of Homo naledi.
Which leaves us with one final lingering thought to mull over. If Homo naledi was indeed actively carrying and burying exclusively their females and young children deep inside this terrifyingly dark, inaccessible cave, what does that say about the emotional lives, the grief, and the societal structures of a species we used to dismiss just because of their small brains?
That is the big question.
What beautiful, complex rituals have been forever swallowed by deep time, hidden in the pitch black?
It's a profound mystery, and one that paleoproteomics is just barely beginning to shed light on.
Thank you for joining us on this deep dive into the sources. Keep asking questions, keep looking closely, and we'll catch you next time as we keep exploring the mysteries of the past.